Behind every breakthrough lies a story, sometimes dramatic, sometimes overlooked, and often scandalous, but always fascinating. I am Dr. Jamie Dixon, and with almost 30 years studying the intricacies of life and the visionaries behind many pivotal discoveries, I take every chance I get to celebrate the scientific past and anticipate its future. Let's explore the hidden history of science together. Our story begins with neurologist Stanley Prusiner. In the early 1980s, he proposed something that challenged the foundations of biology. He claimed that certain fatal brain diseases were caused not by viruses or bacteria, but by proteins. Proteins that could misfold, replicate their shape, and spread that misfolding like an infection and cause diseases like mad cow disease and scrapie in sheep. He called these aberrant proteins prions. The idea was ridiculed because nobody had ever imagined that a protein could be a self-templating pathogen. It violated the central dogma that traits start with a gene, and then the information in that gene is used to make a protein in itself could be viewed as a standalone phenotypic trait. Many scientists resisted, but Prusiner persisted, and he was right. While Prusiner fought for acceptance, another scientist was uncovering a deeper truth. At the NIH, Reed Wickner noticed that certain yeast traits behaved like genes, but didn't follow Mendelian rules. They appeared and disappeared unpredictably. Wickner proposed that these traits were caused by prion-like proteins. One of them was SUP35, a translation termination factor. In its prion form, known as PSI+, SUP35 aggregates and causes ribosomes to read through stop codons, revealing hidden genetic variation. This meant prions weren't just pathological. They might be ancient, functional mechanisms. And this is where this woman, Susan Lindquist, enters the story. A visionary molecular biologist, she believed proteins were far more dynamic than anyone had imagined. She saw folding and misfolding not as accidents, but as opportunities, as switches that could store information. When Wickner proposed that SUP35 was a prion, Lindquist immediately recognized the elegance of the idea. Her lab performed a groundbreaking experiment. They replaced the prion-forming domain of SUP35 with domains from other proteins, and if the chimeric proteins still behave like prions, they reasoned that the candidate must be a prion too. This was the first demonstration that prion domains were modular and transferable. This changed the field forever. Lindquist began to see pretons as adaptive switches, molecular toggles that help organisms survive stress in a reversible manner. Her work showed that pretons could create phenotypic diversity when stress conditions exist and buffer what might otherwise be a rapid population crash, or even worse, extinction. This suggested something profound. Evolution is not driven by DNA alone. Proteins themselves can act as epigenetic elements. But Dr. Lindquist wasn't done. If prions existed in yeast, where else might they be? In 2016, Lindquist's team identified a plant protein, LUMINIDEPENDENS, or LD, that behaved like a prion. LD regulates flowering time in Arabidopsis thaliana. In its normal form, LD helps keep flowering repressed until conditions are right. But in its prion-like aggregated form, LD alters that regulation, allowing the plant to flower earlier. This prion-like state acts as a molecular memory. A plant exposed to stress, like in a drought, can switch LD into its prion-like form, allowing earlier flowering and improving survival. Even more remarkable, this state can be inherited for several generations, but not permanently. It's an ephemeral epigenetic memory, a way for plants to adapt quickly, pass that adaptation to their offspring, and then reset when conditions improve. Lindquist's discovery proved her boldest idea. Prions are ubiquitous, functional, and essential to life's ability to adapt. Susan Lindquist didn't just study prions, she redefined them. She showed that proteins can act as heritable elements. She showed that organisms from yeast to plants use prion-like switches to survive and evolve. Her work bridged molecular biology, genetics, evolution, and biophysics. She saw connections others missed. She believed in ideas long before they were fashionable, and she pursued them with rigor, creativity, and kindness. Sadly, Dr. Susan Lindquist passed away in 2016 at the age of just 67, but her influence continues to shape modern biology today. She taught us that proteins are storytellers, that their shapes and transformations carry meaning. She taught us that evolution is not just written in DNA, but in the dynamic dance of molecules. Those who knew her remember not only her brilliance, but her warmth, a gentle demeanor paired with a visionary mind. Susan was not just a great scientist, but a great person as well, and her discoveries about prions could well lead to revolutions in the way we produce our food and treat disease. Well, there you have it. Dr. Lindquist was a visionary, and like many visionaries before her, she paved paths previously untraveled. Remember that behind every breakthrough lies a story. This was the story of Dr. Susan Lindquist that was fascinating and often overlooked. Her discoveries changed the very foundations of our understanding of biology, but yet most young scientists have never heard of her. I am Dr. Jamie Dixon, and until next time, question, research, and explore.